It's a pleasure to welcome you all to our Discovery and Impact Symposium, our monthly symposium again this month. I'm Darren Johnson. If you don't remember me, I've emceed a few of these over the past year, but haven't for the last couple of months. It's my real pleasure to introduce our first speaker today, Raghuveer Parthasarathy. Raghu is a professor in the Department of Physics at the U of O. He's also a member of the Materials Science Institute, an associate member of the Knight campus, and an associate member of the Institute of Molecular Biology. So you'll see there's some broad interdisciplinary interest in Raghu's talk today. In fact, he got his PhD at the University of Chicago in physics, but I noticed, Raghu, and I took a look at your CV, I hadn't really appreciated, you had a Miller Research Fellowship, which I knew, a very prestigious fellowship at Berkeley, but you actually were, your affiliations listed as Department of Chemistry, and I know you had done a lot of work with Jay Groves, but I had forgotten you formerly were probably a chemist, so we maybe need to get you.
I masqueraded as a chemist for 4 years without anybody catching on, so I just said awesome. Moles once in a while, and yeah.
Oh, you know the secret language even, perfect. So I'm going to start lobbying to our department head that we need to add to your affiliation list to get you co-appointed in chemistry with a courtesy appointment. We should be claiming you too. Raghu's research interests involve biophysics, especially structure and dynamics of the gut microbiome, which his group explores using home-built optical microscopy and image analysis tools. So I think you'll see the topics will fit right in line with the talk. He also has a lot of teaching interests in teaching to our non-science majors, and has for a long time taught classes to our non-scientists, including a current class on biophysics for non-scientists, which I'll appreciate. Not that I'm a non-scientist, but I'm not a biophysicist. So I'm sure I'll get to learn a little extra during your talk, Raghu, and I really look forward to seeing you speak, which I don't think I've seen you give a talk in all of our time at U of O together.
Oh, that's interesting. Cool. Cool. So shall I dive in?
Yeah, the screen is yours.
Fantastic. Cool. Let me start this one up, and thanks for the really nice introduction. Yeah, I'm very happy to be here, and thanks for the invitation. Good. There we go. And let me share this. Oh. Cool. Hopefully you can see a movie playing. I see some nodding. Excellent. Good. So yeah, it's great to be here. And it's interesting. I was looking through the participants list, a lot of people and a lot of names I don't recognize, which is wonderful. So hopefully I can introduce you to some of the things that we've been doing. And in a way, I've packed a lot into this, but there's also, I don't mind not getting to things that I've planned on in this talk. So feel free to jump in with questions. I think Darren had pointed out if you write things into chat, he can see them and then let me know that there's questions up. So, that's all great. So, I thought I'd start by pointing out something that I think this audience is all very well aware of, which is that each of us are actually a whole ecosystem. That each of us is made up of one human and a lot of bacteria, roughly as many bacterial cells as human cells, with hundreds of species in each of us. And nowadays, even the average person on the street knows this also, because we've also learned that this coexistence with these vast microbial communities is really very important to how we function as organisms. And so all sorts of processes related to things like digestion, immunity, early development, and a lot of different diseases, including like complex ones like diabetes, end up being correlated with the composition and nature of this microbiome that we carry along with us. In a way that has attracted a huge amount of attention. So despite this huge amount of attention, there's some very, very basic questions that are quite poorly understood. So we have this community of microbes. What determines the community composition? How can we alter it, for example, to make it have certain species or certain combinations of species, or can we change the abundance of different species? We have a very poor idea of how to do things like this. And it turns out that these questions are extremely difficult. And one thing that my group has been thinking about for a while, could a physical perspective on the microbiome, especially the gut microbiome, which is where most of these things are, could a physical perspective help? So why might one think that that's even remotely a sensible perspective? Well, I'm showing you here a picture, actually it's just from last Saturday. We took a group outing to the coast. And if I were to pan this camera around and pan it around, you'd see this rocky tide pool landscape. And if somebody told you they're going to understand that ecosystem, just by swabbing DNA off of things. And they had no idea that the tides come in and out twice a day, that starfish crawl around, that sea lions can swim in, that there is a rocky topography. You would say, you can swab the DNA, all the DNA you want, but you're never going to really understand this ecosystem. So for macroscopic systems, it's almost self-evident that understanding the ecosystem requires understanding its physical environment. But for the gut microbiome, this complex multi-species environment, nearly everything we know comes from doing things like taking fecal samples and like sequencing them. So extracting like DNA or RNA and doing sequencing to get a sense of what genes are present or being expressed or whatever. And this is immensely powerful. It tells us what species are there, what genes are there, what genes are being expressed, all of that. But it has, excuse me, very poor temporal resolution, depending on how you sample, and no spatial resolution. It gives us, almost no information about the structure and dynamics of this environment. So as a result, we don't know very much about the structure and dynamics of gut microbial communities. And so very kind of basic questions about, are there spatial niches that influence coexistence and competition? What are the timescales of fluctuations, either natural ones or responses to perturbations? How do we think about nucleation, growth, things like that of bacterial colonies? We have really a very limited understanding of any of these sorts of things. Okay, but what can we do about them? Well, what we've been thinking is to answer these sorts of questions. They're the sort of questions that come up in all kinds of sort of biophysical studies. And a route that's very powerful to looking at them is finding a good model system and finding a good way to look at it. So for us, the model system is the zebrafish. And these are a very popular model system for a variety of well-known reasons. They are vertebrates, so they have a lot of physiological similarities to humans and other vertebrates. They are, at young ages, the larvae are transparent or fairly transparent, allowing us to look at them and look at all kinds of processes going on. They're genetically tractable and so amenable to many manipulations. But then also, it turns out that one can treat zebrafish to be initially germ-free, that is devoid of any gut microbes. And then you can add in well-defined microbial groups, including ones you've engineered, for example, to have fluorescent probes and so on. And these so-called gnotobiotic techniques were especially pioneered by our local colleague, Karen Gilliman, with whom a lot of our projects are done in collaboration. So over here is a larval zebrafish. We put a red dye into the gut just for illustration purposes. You can get a sense of what that looks like. So there's our model system, but now we want to image it. Now I said these larval zebrafish are pretty transparent, but it's still challenging to get three-dimensional or to get imaging data from them because we have a fairly stringent set of criteria. We want to image a pretty large field of view that is hundreds of microns on a side. We want to image in 3 dimensions because we expect structure and dynamics to be 3-dimensional in nature. And we need to do this 3-dimensional imaging fairly rapidly because roughly every minute or so, the gut is undergoing these peristaltic contractions, just like your guts are doing as well. So if we image slowly, everything will just be blurred out by those. Plus, we want to be able to image for long periods of time to see dynamics over long timescales. So we need a method that has low phototoxicity or photodamage. So it turns out that there's an optical imaging technique that has these attributes and performs in all of these, in these metrics much better than, for example, confocal microscopy. And it's called light sheet fluorescence microscopy. And I know many of you have heard of this, but don't worry if you haven't. The basic idea is actually a very simple one. What we're going to do is do fluorescence microscopy, but we're going to shape our excitation light into a thin sheet. And that sheet intersects our specimen, our larval zebrafish, here just indicated as a sphere. That sheet intersects our specimen, and then we image with a perpendicular lens everything excited in that plane. So at any instant in time, we get a complete 2-dimensional image, and then we can just scan in the remaining dimension to construct a 3-dimensional image. So we've built actually a couple of home-built light sheet setups now. Nowadays, you can also buy them commercially, and there's interesting things about all of those. But here's our kind of workhorse setup. Each of these are little glass capillaries, And at the end of each capillary, sticking out of the gel in a plug of agar, is a live larval zebrafish. In fact, there's one right there. So our zebrafish are not free swimming, but they're living, they're held in this kind of weak gel so it doesn't constrain their growth. And there they are ready to be imaged. So yeah, any questions about that, about the method? Feel free to jump in if you like. Cool.
Nothing in the chat that I'm seeing, Raghu.
Great, cool. So I'll plunge forward. So what do we see when we do this? So before getting to some of the scientific questions, I'll show you a couple of examples. So here I'm showing you the gut from a fish that was initially devoid of any bacteria. And we've added in, just to the water around it, one bacterial species, which is native to the zebrafish gut and is engineered to express a fluorescent protein. And I'm showing you just one plane over time, and this is in real time. And what you can see is this species, it's like a swarm of bees. Each of those little specks is an individual bacterium. They're just zipping around. There's a little bit of aggregation, but they're mostly zipping around extremely fast. And this is actually what I often tell people, you should imagine your guts are like this. So that's one particular species. Oh, sorry, go ahead. Anyway, other species look different. Here's a different species of bacterium. And here what I'm showing you is a three-dimensional scan through the gut. Some of these bacteria are green, some are red, but they're both the same species. And what you can see is that these bacteria form dense three-dimensional aggregates. So there's a whole range of behaviors, ranging from almost fully individual to almost fully aggregated. These also tend to occupy different locations in the gut. So there is actually this rich spatial structure that we didn't know would be the case when we started looking into it. But it turns out nature is nice to us, and there is this variety of aggregation, spatial structure, and so on. So with this, with having a model system and a way to image it, we've looked at a bunch of things. And almost everything we look at gives us sort of an indication that there are links between physical processes and the sort of activities of gut bacteria. So I'm going to briefly tell you bits of a few stories, and I'm not going to go into a lot of the details because yeah, you can either ask me or look up papers related to the details. The first of these has to do with cholera. And this is especially work from Savannah Logan, a former graduate student of the group. Yeah. So Vibrio cholera is the bacterium that causes the disease cholera. So this is a human pathogen, which still kills about 100,000 people per year. And it's been intensely studied for over a century, but there's still a lot of mysterious things, such as how does it invade a gut? And especially when it invades a human gut, it's not invading some pristine, empty space, but a place that's densely occupied by other bacteria. Now, it turns out that Vibrio cholera has something called a type 6 secretion system, with which it can stab adjacent cells and inject toxins. So we wondered, what is this thing, this type 6 secretion system doing in vivo? So this was a collaboration, especially with Brian Hammer and Xiao Xavier at these places. And Brian Hammer especially has been studying the Vibrio type 6 secretion system and its regulation for a long time. And he's made some interesting strains. For example, ones in which the bacteria is always stabbing and ones in which it cannot stab. And there's others as well. And again, you can details if you want. We thought, okay, what happens if we look at these inside a zebrafish? First, we established that they can actually colonize zebrafish, and I'll skip over that. But what happens if they colonize and there's another species present? So as a simple target species, we picked another zebrafish native bacterium. And it's actually the one I showed you in red and green before. So it has individuals, but it also has large aggregates. So I'm going to show you two movies now. Each of these, we're going to look at this native species, the Eremonas, a day after it's been potentially challenged with one of these Vibrio cholera strains. And what I'm going to show you is a movie scanning through the gut, so a 3D image of what's in the gut. And again, this is the native species. And here's how it looks a day after challenge with the non-stabbing Vibrio cholera. And what you can see is lots of individuals, lots of aggregates. It looks actually like these guys normally look. So it exists at large normal numbers. Here's how it looks a day after being challenged by the always stabbing Vibrio cholera. It's a vast wasteland. So there's like nothing there. Hold on. Oh, sorry, let me pause this. This is the boundaries of the gut. There's a lot of autofluorescence there, which is what you're seeing. But this interior space is devoid of bacteria. If you look carefully, couldn't quite pause it, but there's like 2 bacteria there in the gut. So they're almost completely removed. So we can quantify this. And again, oops, sorry. Invaded by the non-stabbing ones, we have thousands or tens of thousands of the native bacteria. Invaded by the stabbing ones, often we have 0 to 10 bacteria. So this is especially interesting to look at over time. So we can take these 3-dimensional images over periods of like 12 or 16 hours. Here's again the native bacteria invaded by the non-stabbing Vibrio cholera. And there's a rich dynamics that I'm going to come back to later on, but there's always bacteria there. And here, invaded by the stabbing guys, we grow, but then we're very easily just gone. I'll play that one again. So growing, gone over that 16 hours. You can do a lot of quantitative things with this, most of which I'll gloss over. So we spend actually a huge fraction of our time doing image analysis and writing tools to extract data from, or sorry, extract information from these large 3-dimensional data sets, from which you can get things like, what is that population over time, or more elaborate things that I'll come back to later on as well. But again, we see these dramatic, exceptionally sharp drops in the native population when we're invaded by the stabbing bacteria. I'll skip over that. So what is this Vibrio cholera and its type 6 secretion system doing? So our initial thought going in is that, okay, it's stabbing, it's killing the native bacteria. But then we kept looking. And especially we decided to look not just at the bacteria, but at what the gut itself is doing. And this turned up something really quite remarkable. So here we're doing something called differential interference contrast microscopy. And we're just looking at the gut tissue. And we see these peristaltic contractions of the gut. And the image above is from a fish colonized by the non-stabbing Vibrio cholera. The one below is colonized by the stabbing Vibrio cholera. And even by eye, it's really clear that these contractions are much stronger. We have this dramatic increase in the strength of these gut contractions. And we can quantify that by doing things like velocimetry on these images. And this is the amplitude of contractions that we see in a germ-free fish or one colonized by the non-stabbing ones. It's about 100% higher in the ones colonized by the stabbing Vibrio cholera, with outliers that are just enormous in the strength of their contractions. So we're inducing these large gut contractions. And just in the interest of time, I'll skip over this part. It turns out you can ask, well, can you keep the type 6 secretion system but disable its anti-eukaryotic cell activity? The answer is yes. The upshot of all of that is, kind of as illustrated in here most dramatically. Vibrio cholera, can it use its type 6 secretion system to defeat a gut-resident species? Well, the answer very much is yes. We have this enormous drop in the native bacterial abundance, and it seems to be doing that not by direct killing, but rather by inducing strong contractions in the host gut. In other words, by altering the physical environment of the host. And interestingly, the species that's kicked out, like the majority of species that we've looked at, is highly aggregated. So it's very sensitive to these intestinal contractions. In contrast, Vibrio cholera, it's very motile and is quite insensitive to these. So we were excited by this, and it's the first instance that's been found of the type 6 secretion system influencing basically a component of animal physiology. It's usually thought of as like an interbacterial weapon. So there's a lot to look at. You may be wondering, what host cells is Vibrio perturbing to make these gut contractions so strong? And we don't actually know. That's something we're looking at the moment. And we suspect actually macrophages may be the key to this. So macrophages may be key to this. So what one of the graduate students currently in the group is doing is engineering native Vibrio species that have a type 6 secretion system to be able to control this, and monitoring things like macrophage recruitment and death to see if we can uncover these links between the immune system and all of these activities. Here is actually just a fun little rendering of a macrophage in magenta that seems to have engulfed a couple of native bacteria in cyan. So that's one story. Another story that also ends up pointing out some links between physical processes and stuff going on has to do with antibiotics. And this is especially work of these two people, Brendan Schloman, who is a physics grad student in my group, and Travis Wiles, who is a postdoc in Karen Gilman's group. This one is a published one too, although it leads to some things that are pretty cool and new. So it's been known for a while that antibiotics can induce large and long-lasting changes to the gut microbiome. This has been uncovered from like sequencing-based studies. But the especially surprising part is that they can do so even at quite low concentrations, that is sublethal concentrations, such as are often found in the environment. So it's not clear at all, how can this be? How can low antibiotics have a large effect on the gut microbiome? So here again, we thought, can we take our approach of taking a kind of simplified model system and looking at it? So we used a kind of model antibiotic, Cipro, which is widely used. It's a common environmental contaminant as well. And we decided to see, okay, what does it do if you just have one species present? I'm going to show you two different species. One is that very motile one that I showed back at the beginning, and these are zebrafish native species. So if you add a sublethal dose, about a tenth or so of the so-called minimum inhibitory concentration of Cipro to just a flask containing larval zebrafish, you find that it lowers the bacterial abundance in the flask water by about a factor of 10, but lowers the abundance of bacteria in the fish gut by a factor of 100. Okay. So you have a much greater drop in abundance with these low-dose antibiotics than you would expect. But that we expected. What does this look like? Well, here's the movie of these very motile vibrios once they've been treated with this weak antibiotic. And hopefully this comes across reasonably in Zoom. This swarm of bees has turned into a gathering of slugs. I'm not quite sure what the term is, but anyway, they're not zipping around. They're kind of filamented and elongated and are just sluggishly moving. And what happens to these over the course of hours? What happens to these? Come on, there we go. Over the course of hours, is those elongated, slowly moving bacteria are easily pushed out of the gut through these peristaltic contractions and end up being expelled. And we can do staining on these, and these bacteria that are expelled are predominantly live, but yeah, they're no longer present in the gut. Oh, this is popping up that there's a question. Oh, why does the gut look like it splits in half? Oh, that's a good question. Up in this upper image, so what we're looking at is one two-dimensional slice of the gut, which is three-dimensional. I don't know if you can see my hand motions, but there's a lot of structure. There's structure to that. So you have kind of different invaginations and bumps and things like that. So that's one of the reasons that you see what look like bumps or holes. The other is that there are also certain regions where you have a lot of shadowing from the pigment cells that are present. So that's why if you look carefully, you can also see some stripes. Yeah, so we're taking a slice of a somewhat complicated thing. It's not that it's out of focus for those corrugations and stuff. It's like, imagine I had, my hand like this, and I took a slice. My hand isn't a great analogy, but I take a slice in which I have like a little concave part of that full three-dimensional space that'll look like there's sort of a gap there. Plus, if I also have shadowing, which is what that striping is, I could also just lose part of the image because there's no illumination there. Great. Yeah, very good question. There's a lot of subtleties to these images, which I should point out is one of the reasons the image analysis from these can be such a challenge. There's a lot of complexity there. Great questions. Oh, excellent, good. Cool. So for the Vibrio, we have this elongation, and in a way we could have expected this because this filamentation and loss of motility are known from in vitro studies to be common responses to antibiotics. But what it seems to be happening is that the consequences of this are really amplified in the gut because This filamentation leads to aggregation, and there's this coupling between aggregation and transport. So that's a very motile species. But then we thought, let's look at the other extreme. Let's take the most aggregated species that we've imaged so far, this Enterobacter species. It predominantly exists in gigantic clusters. What's it going to do when it experiences weak antibiotics? So I should, well, if this were my class, we would have time for you to chat with your neighbors and make a prediction for what Enterobacter is going to do, and then you have to report back. I guess I'm just going to ask you in your own minds to predict what is Enterobacter going to do with weak antibiotics. And I'll pause for 5 seconds while you make a prediction. So our prediction was, well, it's already aggregated. Who cares? You know, it's not going to care that it might aggregate more. So we thought we'd have less of an effect. But it turns out, in terms of bacterial abundance, we have an even bigger effect. Our concentration or our abundance of bacteria drops by a factor of about 1,000 for these guys. What's going on? Well, when we image this, we notice something really quite striking, which is normally without antibiotic, you have large clusters, but also a lot of small clusters. With the antibiotic, the small clusters largely disappear. So why does that matter? Well, in a nutshell, it leads to a very interesting question just about homeostasis in the gut in general, which is that we know that aggregates can be transported. So how does an aggregating species persist? Well, it has to be always nucleating and growing new aggregates. And so and that's kind of, you can see that in this movie as well. So if you do something that alters that balance of new aggregates coming in, you can totally mess up the steady state abundance of these populations. So can we turn that into a quantitative model? Because I've said that kind of in words. So the answer to that is yes, but before I get to that, let me point out kind of the punchline here, which is that these low-dose antibiotics seem to have a very large effect on things, thanks to this coupling between stress and filamentation and the fact that vertebrate guts are undergoing transport. And these are both very general things. This is what bacteria do in response to antibiotics, and this is what vertebrate intestines do. So it suggests a very general mechanism for antibiotic effects on the gut microbiome. Plus, I'll leave you with this thought that most of the expelled bacteria are alive. So we also are curious to investigate in future things whether this is a mechanism of transmission of antibiotic resistance. Oh, actually, keep an eye. I'm running low on time, so I'm actually going to skip our quantitative model of cluster sizes. But this actually just came out like last week in eLife, so you can read about that more if you want. And instead, what I will do, sorry, let me pause that for just a moment, is give you just a 2-slide glimpse of something. I won't even tell you the full story of that one, but it'll give you a glimpse of another thing we can look at with these, with this system. Okay, so the things I've showed you so far, and don't worry, I'll start the screen sharing in just a moment, all involve watching what the bacteria in the system are doing and watching things like gut contractions and so on. But thanks to the wonders of zebrafish, we can also look at various aspects of the host behavior and things like host response, like immune responses. So what can we learn from that? Just to give you a super brief example, here what we're looking at in magenta are those Vibrio bacteria, the very motile ones I was telling you about. In green are reporters of the so-called TNF-alpha signaling pathway. So TNF-alpha is a cytokine, so it's involved in immune responses. And we see this strong response both in whole regions and in particular cells, like macrophages. Here is the same sort of thing, but we have the same bacteria, but with a defect in their flagella, so they cannot swim. So they don't swim around, and the host has a vastly smaller immune response. We can quantify that. So here's the immune response from the normal bacteria, here the non-motile one, about 2 orders of magnitude smaller. Let's skip all that too, telling us that we can monitor these immune responses, but also in contrast to kind of the textbook picture where the bacteria, where the immune response is just sensing, what's at the cell surface and things like that. The immune response can be manipulated by the behavior of the bacteria, not just their biological or chemical identity. I know that was a little bit fast, but hopefully the general approach comes through. And we can also do this sorts of immune imagings over time and do things like watch groups of immune cells like macrophages zoom around and swarm. Anyway, lots of fun stuff. Let's get that. So in conclusion, hopefully you don't have to remember any of the scientific conclusions, but hopefully I've left in your mind this idea that imaging can actually tell us a lot about gut microbial population dynamics in vivo that you really can't tell or even guess at any other way. And what we find is that these populations are spatially heterogeneous, they're dynamic, and they're influenced by the physical environment of the gut, but they can also influence that physical environment themselves. And yeah, there's a lot to look at here. There's a lot of exciting things for the future. And it's been great working with all the people in my group and also our collaborators, especially down the hall, Karen Gilliman's group. So I know I'm a little bit over, so I'll stop there. And I can't remember now if you asked if question time is now or at the end of both talks, but whatever it is, I'm happy to do it.
Yeah, thanks, Rick. A great talk. And we do have time for a couple of questions now, if anybody has any. And then usually we try and have a little time at the end for just informal discussions if folks can stick around after the hour. So if you're available after 2 for 10 minutes.
Yes,
or so,
yeah, I can stick around. Yes.
Great. Sometimes people will stick around with questions or connections.
Yeah, I'm happy to do both. And I apologize for going a bit over.
No, thanks so much. I appreciated it. So if there are time for a couple questions, I just have the one. Do you think this, were you able to spot, because of a spatial redistribution that you're able to observe in situ in a fish, can you spot things that are not catchable in human? You mentioned fecal testing is kind of the beginning and end of what we have in humans. So you can probably see the same rises and drops in population, but the redistribution piece and all that, is that lost on human analysis?
Yeah, that's a great question. Yeah, sorry, I can't tell who's asking, but that's an excellent
my name's
question.
Dan. I turned on my thing, but
no, great, yeah.
Thanks.
Cool.
Thanks.
So that's a great question. So there's kind of two parts to that answer. One, there definitely are things that you can't tell from looking at fecal samples or even like histology and stuff. The one really key one is this thing of motility. We're finding, as I've shown you some of these examples, some of these species are extremely motile, and that motility can change in response to perturbations like drugs. And that's definitely lost in other things. But the other thing kind of related to that is that we're realizing the timescales for the bacteria changing these things can be vastly quicker than we had guessed. One thing I didn't show you, we have found, for example, one aggregated species that if a different species comes in, it becomes like the aggregates break up and it becomes very like individualistic on a time scale of like less than an hour. And there's no way you'd be able to sort of capture that from other sorts of invasive sampling. And what the deep importance of that is, I'm not sure, but like I think it goes against kind of like the fact that the timescales for changes and things that influence competition can be really fast has been actually really surprising. The other, just to really quickly say, the other thing though that we're trying to do is find things that we can tell by looking that you might be able to map onto things that you can get from like fecal samples and so on. And I didn't have time to talk about it, but like this thing of that I skipped over, which is what are the sizes of clusters or aggregates? Like what is that size distribution? That's something where we can image it in situ and try to understand why it is the way it is, and hopefully say, okay, here are the things to look for in like fecal samples. So it's a great question.
Because what it leads to, sorry, is just that because it really, to me, the why is why should bacteria be able to reversibly cluster and come apart? Because that's a lot of what you've shown, right? So what advantage does it serve them in the environment of our gut and all that? And so, yeah, no,
that's a wonder. Yeah, I think that's a really deep question. Yeah.
I don't know why they would do that, but thank you.
Well, okay, let me give you the one sentence. Okay, I don't know the answer either, but one thing that we've been thinking a lot about is these transitions might be kind of decisions of, do I stay in the gut or do I go try to colonize somebody else? So I'll just put that thought in your mind.
Okay, all right, thank you.
I have a question. This is Scott Clark, and do drugs that alter the mobility of the gut, like Lomotil or something, opiates, how do those affect the population of the bacteria?
Right, that's a great question. We've only looked at that a little bit, and a little bit with drugs, although the more kind of stronger version of that was with like mutants that have like a defective enteric nervous system and just don't have much transport. And we can see, for example, species that wouldn't coexist normally, like do coexist, for example, in ones where we just sort of turn off the transport. So it definitely influences the competition. We haven't kind of systematically looked at that for like a range of different species, but we really only tried like a combination of two. It's a neat question, and I think there's a lot to look at with that.
Okay, I think with that, Raghuveer, since you were able to stay a little later too, maybe folks that have questions can either, your email is easy to find on the U of O website, or can stick around afterwards for questions. But thanks again for a great talk. We'll give you some virtual claps. And
well, thanks a lot. I put my email in the chat too, if people want to.
Perfect.
Feel free to write. Good, thanks.
And with that, I'm going to hand over emceeing duties to Alexia Bashir, who's kindly stepping in for the second half of our session.
Thanks, Darren, and thanks, Raghu, for a great talk. Our next speaker for today is Bhaskar Mandavilli from Thermo Fisher Scientific. He is a senior R&D manager in the biology R&D group. Bhaskar has a PhD in neurobiology from the Department of Biochemistry at the University of Hyderabad in India, and completed a postdoctoral fellowship at the National Institute of Environmental Health in Research Triangle Park in the US, studying mitochondrial DNA damage and repair. He was also a research faculty at the University of North Carolina, studying the role of mitochondria and cardiovascular disease. Bhaskar has been with the company for a little over 15 years in the R&D group. He specializes in high content imaging, toxicology, DNA damage and repair, mitochondrial biology. He's done a broad range of things, as well as developing assays interrogating cell health and death. During his time, he led the development and launch of a variety of fluorescence reagents that cover a diverse range of technology platform, including fluorescent protein-based biosensor, click chemistry, and environmental sensitive dyes. And today, Bhaskar is going to be talking to us about practical considerations when imaging tumor spheroid models and starting kind of to move from a 2D model system to a 3D model system, and what things that we need to take into consideration in that move for research purposes. With that, I'll hand it over to you, Bhaskar.
Thanks, Alexia, for the introduction. And it is a good transition from light sheet microscopy going into the 3D space, because it is the latest instrument that people are using to measure the whole organoids and spheroids, and also whole animal studies using light sheet microscopy and also expansion microscopy. It will be a good transition. Okay, share screen. Okay. Can you see my screen?
Not yet from my end.
Okay.
Something's coming through, yes.
Okay. See all slides. Okay. Now you can see my screen, right?
Yes, we see the PowerPoint presentation.
Mm-hmm.
If you can try to put it in presentation mode.
Yeah, I'm clicking on it, but I don't know what happened. It is not sure. It is not getting into it. It looks like it got frozen or something like that.
Do you want to try like slideshow up top and then take it from there, or is that also not? It's like frozen. Okay.
Try hitting the F5 key, Bhaskar.
Here is F5. Nothing is responding. That is the problem. Yeah, I will let me share it again. Then probably it will help.
Bhaskar, I have a copy of your deck. Do you want me to try to share my screen and then tell me to advance the slides as needed?
Yeah, that should be okay.
Okay.
It worked initially. I don't know what the problem right now.
There's a suggestion to maybe put it in a slideshow mode and then share it. See if that helps. Take it to presentation mode, then share it. I don't know.
I'll wait for you to try that before sharing.
Mhm.
Yeah, I think you want to stop sharing. Yeah, then go to
Yeah,
presentation
yeah,
and then share
and then
again.
share it. Yeah.
Okay. Yeah. Looks like the whole computer froze. Can you present? Can you share?
Yeah, let me
screen?
try.
Yeah, Mike,
Yeah, that's better. Okay, it is just.
get better luck with it.
Sorry about that mess, but yeah, I don't know what is going on.
No problem.
It's the world we live in, right? Everything these days. And then let me swap presenter view. And is everyone seeing Bhaskar slides in presenter view?
Yes.
Looks great.
Yes. Yes.
Okay, great, Bhaskar. Just let me know when to advance.
Thank you.
Yeah, right now I cannot get out of my PowerPoint, so it is my computer is frozen.
Can you see my my screen, Bhaskar?
And just grab your mobile phone and if you need to, just do by voice.
Yeah, that is what I'm going to do because otherwise.
It wouldn't be the
Bhaskar
first
on the
time.
improv here.
Yeah, give me a minute. I will be joining. Can you see the screen now? Yeah, I
We
can.
see Mike's screen.
We
Yeah,
see
yeah, yeah,
Mike's
yeah,
screen.
yeah, yeah.
Can you see it?
Yeah, if you can
Yeah,
see
yeah.
it, Bhaskar,
All right,
then tell
talk
us.
him on through it.
Perfect.
All right, the floor is yours, Bhaskar. So I'll advance whenever you tell me.
Okay, now. Yeah, today I'm going to talk about 3D models and also some practical consideration when you are staining for growing the spheroids, and also when you are some considerations to when you are staining and optimizing the staining of the spheroids, and also some background on imaging and detection and some of the tricks that you can use for high throughput screening of these spheroids as well. So can you go to the next slide, please? So when you consider what is a 3D model, it is basically a cluster of cells that you usually use in 3D, a 2D, or it can be a combination of different varieties of cells differentiated from an embryonic stem cell population, which we call it's organoid. And it can also be extracted from that. The cells can be extracted from a tissue, and using extracellular matrices and hydrogels, you can assemble them into an organoid as well. Next slide, please. And why do people move from 2D to 3D? Basically because it mimics the tissue microenvironment especially when you are doing cancer research. Most of my examples will be related to cancer research here. And then you see cell-to-cell interaction as compared to the 2D monolayer cells. And also there is cell matrix interactions as well. If you are using extracellular matrices or within, sometimes in an organoid tissue, it can be natural matrices as well. And then it enables, keep that on the slide. It also enables you to look at penetration of the drugs that you are using into the organoids or spheroids. And it enables you to look at the drug toxicity in tumor kind of spheroids. And also it enables you to do high throughput screening, either based on imaging or it can be like HTS format as well. So next slide, please. So when you are growing spheroids, we call it a 5-step guide here for creating a uniform cancer spheroids, where you are growing the cells in a monolayer, and then you want to grow them as spheroids. At that point, you want to choose what format you want to use, right? You can grow them in a MatTek dish using hydrogels or extracellular matrices, or the way we do it here to get uniform spheroids is using a ultra low attachment 96 well plate. These are available in 384 well format as well, if you want to more high throughput formats. And then here, when you use ultra low attachment plates, the cells aggregate and form spheroids. And then if you want to do organoids, what we do is we are basically growing stem cells as a ball of cells, and then try to differentiate the cells into whatever tissue or organoid you want to develop it into. And then when you're growing spheroids, another important step is optimizing the cell seeding, depending on what experiments you are doing. For example, if you are doing just kind of drug screening, it is always useful to grow smaller spheroids like 300 to 500 microns. Or if you want to grow them into an organoids over several weeks, it can be a little bit larger, like so it can go up to 1 micron as well. And then once you optimize the cell seeding density and also the size of the spheroid that you want, and how you are growing those spheroids or organoids, the next step will be to characterize to see how healthy your spheroids or organoids are. Right? You do a quick test of size of the spheroid and compactness of the spheroid, and also the cell viability, quick cell viability measurements based on HTS, or you can also use imaging at that point to look at the cell health of the spheroid. Next one, please. So once you know what format or what cell line you want to grow into a spheroid and what format you want to use, then the next step would be labeling and optimization. Here I will show some examples, a series of examples labeling live cells, measuring cell health, and also mitochondrial membrane potentials, and then cell proliferation, and also antibody labeling and some of the immuno-oncology experiments that we did in our lab here using a couple of live cell dyes. And then also last step, I'm going to talk about imaging and analysis. How do you want to just check the health of the size of the spheroid? in a quick fashion, and then also after that, how do you stain with the dyes, and then what are the imaging formats, whether to use wide field microscopy or confocal microscopy. And then also when, as we know, even when you are using confocal microscopy, there is a physical limitation of going deep into the spheroid. You can, after probably 100 microns or 120 microns, usually even with the confocal microscope, it is very hard to get to because of the diffraction of light, the penetration into the deep into the core of the spheroid. So I will show you some methods to overcome that and get more depth and resolution into the spheroid. So next slide, please. So when you are labeling or optimizing the stainings, when compared to the 2D monolayer protocols, there are some differences, mostly as incubation times. Most of the times what we found was you can use the same dye concentration when you move from 2D monolayer protocols to 3D samples. We usually test about up to 400 to 500 micron spheroids. And the only change that we see, depending on what dye you are using, is changing the incubation time. Usually it is longer incubation time. For example, if you are doing a mitochondrial membrane potential dye, usually it is a 30-minute incubation. It is similar to 2D monolayer protocols. And when you are using oxidative stress like CellRox deep red or something like that, where the incubation time can be anywhere between 3 to 4 hours. So it changes between with the dye that you are using. But in general, what we have seen with both antibodies and live cell stains is you need longer incubation times as when you go from 2D to 3D. And then another step is to when you are using drugs, the concentration or EC50s may be different between 2D monolayers and 3D samples. So you may have to optimize the drug concentrations that you are using to create cell death or cytotoxicity in the cells as well. Next slide, please. So when we are talking about labeling and optimization, or when you are, what are you going to study in a 3D sample, right? It is similar to what you do when you are using a 3D monolayer cells. You are using, when you are looking at healthy cell, can you hit the, can you change? It is not the slide change, but to move from one step to another step. So when you are looking at healthy cell, you can look at cell proliferation, mitochondrial membrane potential, and also you can just actually look at intact morphology of the cell using bright field microscopy. And then when the cell is chronically stressed or acute stress, you can measure those by using mitochondrial depolarization dyes, and also you look at oxidative stress or nitrative stress in cells. Sometimes the stress can be reversible, right? Depending on how acute or how high the drug concentration is. During recovery process, you can also measure autophagy or DNA repair enzymes if you want to measure. And if the stress is chronic, it leads to cell death. At that point, it can be necrotic cell death or apoptotic cell death. And in this step, you can measure DNA damage or caspase activation using some of our dyes. And then when you are talking about necrotic cell death, it can be loss of plasma membrane integrity, which we use some of our dead cell dyes to measure those. And then when the cells are dead, you can also measure phagocytosis or inflammatory responses using antibodies or some of our Frodo dyes that we sell. So next slide, please. Here is an example where we used mitochondrial health, measuring high mitochondrial health with using our TMRM dye, which accumulates in live cells based on mitochondrial membrane potential. And also we used CellRox green reagent, which increases upon oxidation of this dye by reactive oxygen species. Here they are U2OS cells treated with 50 micromolar manadione. Manadione is a redox cycler, And when you treat the cells with the menadione, what happens is there is increase in oxidative stress, and the mitochondria are stressed out. So you see decrease in mitochondrial membrane potential, as is seen with the decrease in staining of the dye with TMRM and increase in cell rocks dye staining. So next slide, please. Next slide. Here we are going to measure caspase activation in combination with mitochondrial depolarization. Here is where we grew spheroids. These are about 400 micron spheroids in an AU bottom nung plates. And then these were grown over 24 hours. You plate the cells, these are ultra low attachment plates. You form compact spheroids. These are A549 cells here. And then after growing for 24 hours, we just do use different concentration of niclosamide drug. Highest concentration is 40 micromolar. As you can see, as you increase the concentration of niclosamide, there is decrease in mitochondrial membrane potential and increase in apoptotic staining. So using our high content imaging systems, you can actually segment the whole spheroid in the bright field as one object and quantitate the fluorescence within that spheroid. As you can see on the graph here, you can measure the intensity of mitotracker orange signal and also cell event caspase 3/7 signal. As you can see, as the mitochondrial membrane potential goes down, there is increase in apoptotic signal. Next slide, please. So next I'm going to show how to measure cell proliferation within the spheroid. This is using our click chemistry, which you probably already know. I'm not going to spend too much time on this. Is basically azide alkyne reaction. So you conjugate one of the molecules to azide and a fluorescence molecule to alkyne, and then you do click reaction to get fluorescence in the cells. So next slide, please. So here I'm going to show just comparison of the protocols, the traditional BRDU protocol against the EDU protocol using click chemistry. So next slide, next, click on that. And then when you, basically what you do is you incubate either BRDU or EDU with the cells. There is BRDU incorporation into the DNA of the proliferating cells. And then you go ahead and then DNA is digested to get the antigen retrieval, and then use BRDU antibody to create fluorescence in the proliferating cells. So this requires acid DNase digestion for BRDU protocols, which takes longer time when you compare it to the EDU protocols. Just keep clicking because we are short on time. Yeah. Then when you do EDU, it's similarly incorporated into the proliferating cells DNA, and then the further downstream process is much shorter when compared to the BRDU protocol. You do a click you fix the cells, permeabilize the cells, and do a click reaction using a fluorophore conjugated alkyne, this azide, and then create fluorescence. So here we did click reaction. As you can see in green, all proliferating cells within the spheroid are highlighted here. And then it takes 30 minutes of incubation. There is no change between 2D versus 3D. And then we do Ka67 antibody staining after we do click reaction. When you do antibody staining, the protocols are a little bit different because you include 5% DMSO and heparin when you are incubating with both primary and secondary antibody to reduce some of the background staining. And DMSO is to actually facilitate the penetration of the antibody into the core of the spheroids. So following these protocols, we get good staining with both Ka67 and also EDU staining to look at the proliferating cells. And when you incubate with 50 micromolar hydroxyurea, which is a inhibition which inhibits the DNA synthesis in the cells, you can see complete inhibition of the EDU incorporation into the spheroid, the cells of the spheroids. These are HeLa cell spheroids. And again, by using our high content imaging system, you can actually segment the whole spheroids as a single object, and then you can count the number of EDU positive cells or look at the whole intensity within the spheroid to look at the inhibition of EDU positive cells in hydroxyurea treated populations. So as we move along this pathway of cell health and death, the next one will be loss of plasma membrane integrity, usually during necrotic cell death or late apoptosis. Here I'm going to show a couple of models, immuno-oncology models, where we either stain it with, treat it with antibody drug conjugates, here using our site-click antibody conjugation kits. Here what we do is the unconjugated antibody is modified using GalNAs to create galactosidase activated antibodies. And then after using our S-dibo Frodo Red, we conjugate both the dye and also the drug to this antibody. So when you incubate the spheroids, these are SKBR3 breast cancer spheroids. When you incubate with drug or without drug, these spheroids, as you can see on the left side, when you don't use the drug, there is not much cytotoxicity. The spheroids are intact. What you see is only the Frodo incorporation there. And when you have the drug itself, you can see there is a lot of cytotoxicity, there is a lot of dead cells, as you can see with the cell event Caspase 3/7 sensor. This sensor is basically a DVD substrate, which is Caspase 3 specific, that is conjugated to a green dye. When the caspases are activated, it cleaves the DVD peptide, and then the fluorophore is released and it binds the DNA in the apoptotic positive cells. As you can see here, there's a lot of positive green cells in here because of the toxicity of antibody drug conjugates. Next slide, please. Here is another example where we did it in live cells again. These are HeLa spheroids grown on a U-bottom nunc ultra-low attachment plates. Here, what we did is instead of antibody drug conjugates, we isolated NK cells from blood samples, activated them, and then added to the spheroids. As you can see on the left 3 columns, there are no NK cells, there is only drug. Cetuximab drug is a EGFR receptor-based drug, which activates the killing of the cells. And with NK cells, and then last 3 columns are NK cells plus cetuximab drug. These are incubated for 6 hours. Can you keep that on the previous slide, please? So as you can see in this population of the spheroids is with just NK cells, you don't see any. Here the NK cells are stained with cell tracker deep red, so that you will know when they are entering the spheroids, and also to track them within the spheroids. And then what we use the cell event Caspase 3/7 sensor here to look at the cell death within the spheroid. And as you can see, there are no, when you add NK cells, there is some cell death, a little bit more than what you see in the control population. And when you add NK cells along with the drug, you see a lot more cytotoxicity and also apoptotic cell death. So next slide, please. Again, here we quantitated this using segmenting the whole spheroid as a one object, and then looking at the mean signal intensities of the cell event Caspase probe. As you can see here, you see increasing number of apoptotic cells within the spheroid as you go from NK cells and then antibody plus NK cells, which means that antibody is potentiating the NK cell mediated cell death as seen by the increase in apoptotic cells. Next slide, please. So when you are looking at downstream of imaging your stained spheroids, you can use wide field microscopy to just check your spheroids before staining or after staining. to look at how the staining is. But if you want to look deep into the spheroid, you have to, wide field doesn't work because of the diffraction of light, and then you may see hazy pictures of after a couple of layers within the spheroid. And when you use, can you click on, yeah, when you use confocal mode, there is less of diffraction, there is more focal light on the sample, you can see more resolution, and also you can count more number of cells within the spheroid. Can you go to the next slide, please? Here is an example. Even when you go to from wide field to confocal mode, when the size of the spheroid is like 400 to 500 microns, it is very, very hard to get depth into the spheroid after 180 to 100 microns without, because there is refractive index mismatch between the sample and the objective. And to overcome some of those issues, we clear the samples. One of the examples we give here is the cytovista clearing agent, which diffuses into the spheroid and matches the refractive index that with the objective that you use for the confocal microscope. So there is more you get see more resolution and also more depth into the spheroid. So you can count more number of cells within the spheroid using clearing reagent. Can you go to the next slide, please? So here is an example where we grew, this is a data from one of our collaborators. They grew different sizes of the spheroids. As you can see, as you increase the size of the spheroid, without clearing, there are more dark areas within the core of the spheroid. And when you clear this with the clearing reagent, you can see some of those dark areas can be measured using the confocal microscope. Next slide, please. So next slide. So here is an example where I'm just talking, where you can use normal wide field microscope to just look at your cells, how the spheroids are across the plate. This is an example of the 384 well ultra low attachment plate, where you can see the spheroids are intact across the plate in the 384 well plate. So yeah, next one. And then using our, if you want to get more penetration into the spheroid, we use high content imaging instrument using confocal mode. Using this, you can use Z sections across the spheroid, where you can get more individual Z slices, and you can quantitate in the individual Z slices, or you can project it as a maximum intensity projection and get overall intensities within the spheroids. Using this, you can actually measure 7 different colors within the spheroid. So you can measure 7 different markers within the spheroid using this instrument. Next slide, please. So here is an example where the couple of other markers were used. This is a 3-plex assay, Hoechst, and also the couple of other markers. As you go from bright field, you can actually see in 4x, you just segment the spheroid as a single object using bright field, and then you can look at the intensities of the markers within the cells using the spheroid as an object. Next slide, please. And also this instrument provides you a smart scanning. One of the problems we have when you grow them in a 96 well format is the spheroids are located at different positions within the well of the 96 well plate. So when you are measuring in an automated format, what happens is when you are scanning the plate, they may be at the different locations within the plate. You end up scanning the entire well, which may take you about like 5 to 6 hours when you are scanning the entire well to go through the 96 well plate. So what we did was like we created smart scanning module within the imaging software, where you can use a 4x or 2x magnification and look at which covers the entire well in a single field of view, and then you actually segment that in a lower magnification And when you go to a second scan, what happens is it will go to that particular field where the spheroid is, and you can do higher magnification scanning at that particular time. So when you compare with 5 to 6 hours using our smart scanning, you can actually do the entire 96 cell plate in about 28 minutes.
Bhaskar, just
Yeah.
a quick reminder, we're like 10 minutes past the hour. I think
Yeah,
we
yeah.
have 15 minutes, so if
Yeah, this
around
is
5 to 6 minutes.
this is the last slide, so summary
Okay.
slide.
Great.
So as what we have seen in last few slides is that when you go from 2D to 3D, the concentration of dyes mostly stay the same. It is mostly the incubation times and some of the, when you are using antibody protocols, you may have to include some additional reagents like Twin20 or heparin to reduce the backgrounds, or DMSO to increase the penetration of your molecules into the core of the spheroid. And as I said, and to increase the signal to background when you are using antibody staining protocols. And also we also talked about some of the strategies that you need to use, confocal, and also some clearing reagents. As for clearing reagents, a lot of other clearing reagents as well that people use. I showed one of the clearing reagents that we sell, and to improve the resolution and also the depth of imaging when you are doing larger spheroids. Thank you for your attention, and also sorry we wasted 5 minutes initially because we had some problems with them.
Oh, no problem.
Yeah.
Thank you very much, Bhaskar, for overviewing, the reagent toolkit as well as some imaging technologies and instrumentation that comes handy for 3D model studies. And thanks, Mike, for helping us with the technical challenges. Any questions? Like, we have a couple of minutes. Any questions from the audience for Bhaskar?
I'll just first a comment, then followed by a question. But both of these talks showed the critical importance of spatial resolution in your measurement, right? Neither of these could be done by bulk biochemical, or even if you manage to do flow effectively with 3D cultures, which is what I want you to comment on. Why not do flow? But the main reason I can think of is why both these talks are around is that you'd need the spatial relationship as well as the dynamic on-off of the sensor systems to catch. So they're going to remain imaging paradigms, right? Or why not sort spheroids by flow? Is it just physical because they're too big? So
because
zebrafish?
As you said, it one of the problems using flow is you don't get spatial resolution, right? And also the workflows become more difficult because you have to use more markers to delineate what is in the core of your spheroid and what is in the peripheral layers, right? So if you can get the depth into the spheroid, it is much easier to correlate what exactly happens when compared to a tissue microenvironment as compared to flow workflows. Does it answer your question, Dan?
Yeah, it does. I was really just kind of bringing up, I was looking for some commentary on your part about the spatial importance, or the importance,
the
rather,
spatial importance is
piece.
very important because
sometimes
because of cell interactions, and also it gives you a more in-depth view of actual tissue microenvironment. Yep.
I have one question for you, Bhaskar. Are we seeing more and more adoption of 3D cell models by customers and researchers with the development of maybe new reagents, these optimized protocols, new imaging modalities? Or are you seeing the adoption a little bit still slow? And why would that be if that's the case?
Yeah, the adoption of 3D models is it is growing fast. The problem with detection is still present because of the problems I talked about. Even if you use confocal with clearing reagents, it's still getting depth into a very tight organoid is still a problem, because with spheroids, it is easier because they're not as compact. So when you are growing like a mini tissue or organoids, they're much more compact, and then getting light into the sample is very, very difficult. That is where that probably the clearing in combination with the expansion microscopy and light sheet microscopy comes in to get more depth into the sample. So the technology is still developing in terms of light sheet and other modalities. So it will take some time for people to catch up, I think.
Yeah, I agree, Bhaskar. I think that it's important, and it's really highlighted also by Raghu's talk, where the idea of more and more physiologically relevant systems. I mean, obviously working in vivo brings up some serious channels in terms of technique. But even in models like you showed with tumor spheroids, it's important to keep in mind just environmental conditions, right? Oxygen tension and such. And even these models don't have vascularization, right? So to me, the sort of nascent frontier that might be a nice balance between whole animal imaging and models that are 3D, like spheroids, are organoids. And I think that we're really at the nascent stages of understanding, both with respect to model development and protocols for using sensors in these kind of more difficult systems. Not only regarding the sensors, but also regarding just the conditions, imaging conditions under which we might be able to interrogate them effectively. So I think this is really interesting to have these two talks in the same session, because it kind of points at two parts of a problem that Dan highlighted in really needing physiological relevance, models that are not just single cells, and really start to get at some deeper questions. So thanks to you both. I enjoyed both talks.
Yeah, it will be. I'll be interested in connecting with Raghu and then see what we can do to solve some of the issues we have with imaging compact organoids or spheroids. Yep.
Thank you very much. There's some commentary in the chat, if you haven't checked it out, regarding a question from David to Raghu regarding the use of sensors to kind of interrogate the molecular kind of mechanisms. And Raghu said, we need some help with that. So this is a great opportunity for some follow-up discussions around that and kind of determining some paths for collaboration on that front.
Yeah.
Sensor development, as well as kind of leveraging the imaging expertise close by for some 3D model work. Thank you everyone for attending. Thank you for two great talks and for sticking around with us extra 20 minutes. Thank you. Have a good afternoon.
Thank you.
Take care.
Thanks so much.
Thanks a lot. This was fun.
Bye. Thank you.
Bye.